Psalm 118, verse 24. The Lord has done it this very day. Let us rejoice today and be glad. Now that we know our somatosensory receptors and afferents, we're ready to learn about the entire somatosensory pathway, bringing information from our somatosensory receptors to the central nervous system and up to the brainstem, cerebellum, or cerebrum for additional processing and awareness. We're going to start with an overview of the somatosensory pathways. Abbreviations in this part of our lecture include ANS, autonomic nervous system, SNS, sympathetic nervous system, PSNS, parasympathetic nervous system, CNS, central nervous system, T, thoracic, L, lumbar, and S, sacral. The sensory system provides our brain with information about our environment, both the external environment, what is going on around us, as well as our internal environment within our bodies. Sensory information is a nerve impulse that is generated from a stimulus. Perception is when we become aware of that stimulus. We then must interpret the stimulus to determine what it is. Perception and interpretation occur in different parts of the brain. Somatosensory pathways begin with the somatosensory receptor. We have learned about several different types of somatosensory receptors. These somatosensory receptors are specialized endings of pseudounipolar neurons. They are located at the ends of the peripheral process of these neurons. Because these neurons are carrying information from the periphery to the central nervous system, they are known as afferent neurons. And because these particular neurons are the first neuron in a series of neurons, they are also called first-order neurons. Somatosensory receptors are essentially biotransducers. That means they convert some form of a stimulus to an action potential. They do so by opening up modality-gated ion channels, allowing the influx of ions into the afferent. This creates receptor potentials. If we have sufficient summation of our receptor potentials, we open up voltage-gated sodium channels and generate action potentials. To summarize, the somatosensory information is going to start at the receptor and then in the afferent in a peripheral nerve where it will be carried to the central nervous system. It will then travel along neurons in somatosensory tracts, carrying the information to the target location within the central nervous system. The target location could be nuclei within the brainstem, it could be the cerebellum, or it could be target areas within the cerebrum. When it comes to the sensory pathways, I would like you to know the location of the tracts that we discuss in class and the relations to each other. So which one is anterior to another one? Is it situated more medially? Is it situated more laterally? I want you to have a good concept of where these are located because that makes a huge impact when it comes to different injuries as far as which tracts are damaged and which tracts are spared. Keep in mind that white matter regions contain axons, and that axons that have similar function tend to travel together in tracts or fasciculi. You can think of those as just two names for a similar kind of thing. And then we have tracts that can be sequentially linked together. So this tract is going to go to the next tract, and that will often be called a system. If a pathway is disrupted at any place between the receptor and its target in the CNS, there's going to have some alteration of perception of the sensation that would have been carried in that pathway. Where we experience this altered sensation is going to depend upon where in the pathway the disruption takes place. Here we have an image showing a typical spinal cord segment. Although the spinal cord actually looks different in the different regions, we're going to use just a typical general segment in this course. This image also shows the tracts that are located within the spinal cord. Blue tracts are sensory tracts, and red tracts are motor tracts. This week, we're going to be learning the blue tracts, and there are 3 of these tracts that you will need to know the names of and the location within the spinal cord. Starting in the far back, we have the dorsal column. Note that in this drawing, it is labeled as dorsal column medial lemniscus system. But you will recall that a system is actually a series of tracts linked together. In this case, one tract in the spinal cord called the dorsal column, and a second tract in the brainstem called the medial lemniscus. So make sure when you're in the spinal cord that you realize that you are not looking at the dorsal column medial lemniscus system, you are simply looking at the dorsal column. The dorsal column contains within it 2 different fasciculi, the gracile and cuneate fasciculi. But we are only going to discuss the dorsal column as a whole. You will probably never see the gracile or cuneate fasciculi in the clinic, in any kind of medical reports. So we're just going to focus on the dorsal column. As we move along anteriorly and staying along the periphery of the spinal cord, we come to the next blue tract that I want you to know, and that is the posterior spinocerebellar tract. There is an anterior spinocerebellar tract, although we're not going to talk about that particular tract much in this class. The one I want you to know is the posterior spinocerebellar tract. As we continue to move anteriorly and now a bit medially, wrapping around the ventral horn, we have our 3rd sensory tract that I'd like you to know, the spinothalamic tract. There are 2 separate parts to the spinothalamic tract. There's the lateral spinothalamic and the anterior spinothalamic. But functionally, there is so much overlap between what is in these 2 parts, we're just going to put them together and just know the spinothalamic tract. Next week, we're going to move into the motor system. There you will need to know one of the red tracts. So to get a head start, I've circled it for you here. It is the lateral corticospinal tract. Here is a blank image of a spinal cord segment that we will use frequently throughout the course. If I were to give you an image such as this one, you should be able to draw in the different tracts that we just discussed. So for this week, you should be able to draw in and label on this image the dorsal column, the posterior spinocerebellar tract, and the spinothalamic tract. Next week, you should be able to add in the lateral corticospinal tract. The gray matter of the spinal cord is broken up into subdivisions. These subdivisions are known as Rexid's laminae. They're basically functional subdivisions, and they are histologically different from each other. Here you see a picture of the gray matter of the spinal cord with Rexid's laminae shown on the left-hand side. You can see how we have 10 different sections, Roman numerals 1 through 10. And they essentially go from the dorsal horn, they go all the way down into the ventral horn, and then around the central canal. In the next few slides, I'll go over the different laminae in a little bit more detail, but I'm not going to ask you to know them to any great extent. More big picture. The dorsal horn is made up of the first 6 laminae. So our sensory input that's coming in from the periphery is going to be going into the dorsal horn in order to synapse onto second-order neurons and interneurons. You will not need to know the specifics about these different laminae. Just know dorsal horn is made up of laminae 1 through 6. The ventral horn is made up of laminae 8 and 9. You'll recall the ventral horn is the location of the somas of our voluntary motor neurons. There are also somas of a variety of interneurons located in this region. Lamina 7 is situated in an area that is in between the dorsal horn and the ventral horn, an area we call the intermediate zone. There are a couple of different structures that will be in lamina 7 that are important to know. The first is the nucleus, known as Clark's nucleus. Clark's nucleus contains cell bodies of second-order neurons that are going to be carrying unconscious proprioception information from our lower extremities and the majority of our trunk all the way up to the cerebellum. The axons of the neurons, whose cell bodies are in Clark's nucleus, run in the posterior spinocerebellar tract. We'll talk about this pathway more later on in our lecture. Lamina 7 is also the location for the cell bodies of our presynaptic autonomic nervous system neurons. Between T1 and L2 or L3, we have a true lateral horn, and this is the location of the presynaptic sympathetic neuron cell bodies. At S2 to S4 spinal cord segments, we do not have a true lateral horn, but still within lamina 7, we have the presynaptic cell bodies, this time for the parasympathetic nervous system. And then finally we have lamina 10, which is simply the zone surrounding the central canal and is made up mostly of interneuron axons as they are crossing from one side of the spinal cord to another.